Good evening to my respected teachers and fellow students. I'm Bishnupriya, a student of Durgapur Government College and from SEM 2. And today I'll be delving into the fascinating world of computational analysis of bioactivity, focusing specially on citrulline and tannin. Let's explore how computational methods can revolutionize our understanding of their bioactivity and pave the way for innovative medical applications. Next slide, ma'am. So my topic of the presentation is studying the bioactivity of citrulline and tannin through computation. The next slide is about the contents. So these are the contents which I will share one by one. Firstly, introduction, second, objectives of the project, then methodologies used, then main findings, then conclusion, and at the last, references. Next slide. So first of all, the question is about what is bioactivity. Bioactivity refers to the ability of a substance to exert a biological effect on living organisms. This effect could be therapeutic, such as a drug interacting with a target receptor to treat a disease, or it could be harmful, such as toxin affecting cellular function. Next. So I have chosen citrulline and tannin for as a bioactive compound. Mainly I have chosen L-citrulline and hydrolyzable tannin. So the uses of L-citrulline is enhanced athletic performance, increased nitric oxide production, improved muscle recovery, regulation of blood pressure, potential benefits of erectile dysfunction, and ammonia detoxification. The uses of tannins are leather tanning, medicine, in food additives, wine making, wood preservation, textile dyeing, cosmetics. So now let's explore the next point, objectives. The objectives of the presentation are we have used ChemDraw and Pass Online to analyze the bioactivity of the chosen compounds. We have done the literature survey of these compounds and explained them in this presentation. We found the following useful bioactivities of the compound from Pass Online website. I have found the following bioactivities for my compounds. For citrulline, these are antitoxic, antihypertensive, metabolic disease treatment, nootropic, growth hormone agonist. For tannin, the bioactivities are antioxidant, keratolytic, antiseptic, antihypercholesterolemic, and astringent. Now coming to the methodology, so which methods I've used. Firstly, we have used ChemDraw to draw our chosen structure and save them as MDL mol file. Secondly, we have registered ourselves in the Way to Drug Pass Online website and predicted the bioactivity of the chosen compounds. Next, we did a literature survey for which the results has been explained in the next section. So these are nothing but the screenshots of the methods I have used. Next slide, ma'am. So now moving on, main findings. The bioactivity of citrulline. As antitoxic, citrulline is an amino acid involved in urea cycle detoxification. It's not inherently antitoxic, but it plays a role in removing toxic ammonia from the body by converting it into urea, which is then excreted. Next is antihypertensive. Citrulline may have antihypertensive effects by promoting the production of nitric oxide, which helps relax blood vessels. Next is metabolic disease treatment. Citrulline shows promise in treating metabolic diseases due to its role in improving vascular function, insulin sensitivity. It also helps regulate amino acid metabolism and may aid in reducing reducing inflammation, which are all factors in metabolic disorders like diabetes and obesity. The next topic is nootropics. Nootropics are a diverse group of medical substances whose action improves human thinking, learning, and memory. Nootropic, also known as cognitive enhancers, are drugs that some people use in an attempt to improve memory. Some research suggests that citrulline may indirectly support cognitive function by improving blood flow and nutrient delivery to the brain due to its role in promoting nitric oxide production. Growth hormone agonist. Citrulline has shown potential as a growth hormone agonist, meaning it may stimulate the release of growth hormone in the body. This could have implications for muscle growth, recovery, and overall physical performance. Bioactivities for tannins are antioxidant. Tannins are a type of polyphenol compounds found in plants. They have antioxidant properties, meaning they can help protect cells from damage caused by free radicals, potentially reducing the risk of chronic diseases such as cancer and cardiovascular disease. Next, ma'am. The next bioactivity is keratolytic. Keratolytic agents help remove dead skin cells and promote exfoliation. Tannins have astringent properties that can help tighten and tone the skin, but their direct keratolytic effects are limited. Next is antiseptic. Tannins possesses antiseptic properties, making them effective in inhibiting the growth of bacteria and fungi. They can be used in various applications such as wound care, oral hygiene products, and topical treatments for skin conditions like acne. Anti-hypercholesterolemic. Tannin may lower cholesterol levels in the blood. Some research suggests that certain tannins can interfere with the absorption of dietary cholesterol in the intestines, leading to reduced levels of cholesterol in the bloodstream. The last bioactivity of tannin is astringent. Astringent tannins cause a tightening in the mouth. They are found in foods like tea, wine, and some fruits, contributing to their characteristic taste and mouthfeel. Before going to the next slide, I would like to say a very important point that if we intake citrulline and tannin excessively, what will happen? Excessive intake of tannin can cause gastrointestinal issues like diarrhea, stomach cramps, lower blood pressure, etc. And excessive intake of tannins can cause digestive issues, constipation, etc. To avoid these risks, it's important to follow recommended dosage. So the next the next slide is about the conclusion. Computational bioactivity studies provide valuable tools for understanding the multifaceted roles of citrulline and tannin in biological systems. By leveraging computational modeling and simulation techniques, researchers can uncover new insights into citrulline and tannins mechanism of action, predict their biological activities, and inform the development of therapeutic interventions targeting citrulline and tannin related pathways. The next slide is about the references which I have used to complete this presentation. I have used PubChem, ChatGPT, Nimbus platform, and Google. So thank you all for your attention throughout this presentation. I'm grateful for the opportunity to share my research with such an esteemed audience. I look forward to explore new avenues of discovery